Okay, so if you're watching the recording of this, I'm going to go ahead and start now, even though I don't have anybody else on here, because I don't want to sit here for too long waiting for participants. So we're going to go ahead and get started. So if you guys are going back and rewatching this, you kind of know what happened here. So we're going to talk about chapter 23 with our GI diseases. We'll recap that. We'll recap chapter 24 with our genitourinary diseases. Then we'll talk about the most missed topics in those. And I'll field any questions about these chapters if you have any. And then after that, we will get into our review for the units. So starting off with chapter 23, we talked about our diseases of the gastrointestinal system. Remember that dental caries is definitely like our number one as far as what the most common infectious disease is in human beings. So we're talking about tooth decay here. Usually
caused by biofilms formed by the microbiota of the mouth. And depending on what your microbiome might be, can put you at higher risk for having these issues. So that could even lead to the development of trench mouth if you become exposed to the bacteria that are associated with trench mouth, also known as necrotizing ulcerative gingivitis or periodontitis. Remember too that as we get into the GI stuff, your gut health is important for more than just for your like actual gut function. We're also talking about, why can't I zoom on this? Here we go. We're also talking about the microbiome of your gut being important for training your immune system and things like that. Right. So then we have gastritis, which we talked about. Those are gastric ulcers. So gastritis, itis meaning inflammation. Gastro is talking about your stomach here. So inflammation in the stomach caused by the presence of the bacterium Helicobacter pylori. We also have various causes of diarrhea, and what we need to remember about those, that could be caused by these bacteria Shigella, which has the Shiga toxin and enterotoxins that cause damage in the intestine, and that can lead to the presentation of dysentery, which is bloody diarrhea. And then we have, which is something that isn't necessarily on our study sheet here for dysentery. I don't know why it's not on here. That's okay, but it's not on your exam. Hey, I've already double checked it. So no worries, but you should probably know that term for medical reasons. I'm just saying. So Shigella, Salmonella, Campylobacter, S-Tech E. coli, which is our Shiga toxin producing E. coli, and then C. diff, which is the only one that's gram-positive. We know it's gram-positive because it's Clostridioides, or it used to be Clostridium difficile. This guy's an endospore former, and all of our endospore formers are gram-positive. And then we have our antigens on their surface, whether they have flagella, whether they have capsules, and whether they have certain markers on their surfaces like LPS that help identify these guys. Those are our HOK antigens. Diarrhea is best treated with fluids and electrolyte replacement. That's an obvious one that we know, but it is in fact one of those facts that we need to know for the course. Cholera, this is caused by the bacterium Vibrio cholerae. This is diarrhea causing as well, but this leads to a very characteristic presentation with the rice water stools, causing the toxins that it produces, the cholera toxins that it produces, cause really massive electrolyte release, leading to serious, potentially deadly dehydration. Cryptosporidium is a diarrhea-causing protozoa. The cyst of this protozoa is very resistant to chlorine treatments. We often see this in swimming pools. Norovirus and rotavirus are common diarrhea-causing viruses. There's a vaccine available for rotavirus, and it's probably the most common cause of diarrhea in general. Norovirus is most closely linked with cruise ship outbreaks of diarrhea, but it can be found outside of the cruise ship environment as well. Food poisoning, here we have toxins that are produced by certain bacteria that cause this intoxication or poisoning effect. You are not having an issue with infection from these guys, but you are having a reaction to the toxins that they've produced. For our chronic diarrheas, we had EAEC, which is enteroaggregative E. coli, and then protozoa, Cyclospora, Giardia, and Entamoeba that can also lead to chronic diarrhea or dysentery. Then we had hepatitis, which is inflammation of our liver. And we talked about basically the different forms of that. We have hepatitis A, which is our fecal oral route. Hep B, which is blood and sexual contact. We do have vaccines for A and B, but we don't have treatment available for B. Usually a lot of times with A, you'll just get over it on your own. And then C, you have to have proper blood transmission involved with this. Let me get out of this for a second. Here we go. Yeah. So I can see the rest of my helmets down here. Anyways, C, we have proper blood transmission, so it's a little bit more involved as far as exposure to the blood. And then there is no vaccine for that one, but there is treatment available as long as it hasn't progressed into a more chronic phase. Then we have, and so you can clear out hepatitis C infection with medications. It is curable, potentially. Then we talked about our helminth infections. Remember, helminths they are part of Animalia, kingdom Animalia. They're multicellular. They have different stages in their life cycles, eggs and larvae, even different stages of larvae. Some of them have intermediate hosts involved. Ultimately, the host that we are actually reproducing in is called the definitive host. Anyways, we talked about pinworms, which are the ones that were causing the anal itching. This was caused by Enterobius vermicularis. We had whipworms, Trichuris trichuria. This is from contaminated, fecally or waste contaminated like soil. Then these worms, the larvae can get in through the intact skin and migrate into the system that way. And you'll pass the eggs through your feces. Then we have Ascaris lumbricoides, the excuse me, the giant intestinal roundworm. These guys being roundworms, right? So this is going to be nematoda. These guys are the most common cause of helminth infections across the world. Tapeworms, we had various different ones. We have Diphyllobothrium latum, it's associated with sushi. Hymenolapsus, which is like our rat tapeworm, which is the most common tapeworm across the world. And then Taenia solium, which is our segmented flatworm tapeworm, the one that we're most used to talking about, the one that can be like 5 meters long. They have these segments called proglottids that break off that have eggs inside of them. If you ingest contaminated meat, you're more likely to get the true GI symptoms associated with and the true GI presentation of the tapeworm. Whereas if you ingest the proglottids directly, you're at risk for developing cysticercosis. And if that's in your brain, that's neurocysticercosis, where we have this incystation of the larvae in the brain, that if their cysts are broken open, like during surgery to try to remove them, the inflammation that can result can be deadly very, very quickly. Then we have the Chinese liver flukes, Opisthochus sinensis, and Clonorchus sinensis that can potentially block the bile duct and cause serious issues with the liver. And then we have trichinosis. This is trichinella helminths. These guys are going to find their ways into the muscle tissue and insist in the muscle tissue. And then you eat undercooked pork that can be transmitted to you that way. And then they can move into your muscle tissue and cause diffuse inflammation throughout the body, and including damage to tissues from that inflammation. Then we have schistosomes. These are our blood flukes. And we have 3 different strains that we kind of learned about in class. 2 of them, Masoni and Japonicum. These guys are usually associated with our typical blood-based and overall tissue-based blood fluke presentation. These guys get in through your intact skin from contaminated water and migrate in, causing swimmer's itch, how they get in. There is a intermediate host for these guys, the snail. So it's also called like the snail fluke. But so those will migrate in through the skin and get into other tissues of the body. It can cause splenomegaly, hepatomegaly, and all sorts of presentation of inflammation of tissues in the body. They can coat themselves in antigens from the host's bloodstream to hide away from the immune system, which is a pretty cool way to shield themselves. There's another one, schistosoma hematobium, that is associated with bladder infections that we learned about in the next chapter after this one. So that can cause inflammation in the bladder, bloody urine, as well as complete like blockage in the bladder as well. Chapter 24, this is where we talked about our genitourinary diseases. With our colonization tends to be pretty similar, with lactobacillus being very predominant in both female and male. We can also have presentation with Candida albicans, which is a fungus, a yeast, right? And yeast are fungi that are part of our normal flora. Our flora is going to change due to hormone changes, either throughout the lifetime or even from our monthly cycles. With men, that doesn't change as much from hormone fluctuations, since they don't have monthly cycles and stuff they're subjected to. So they don't have quite the same shifts there, but the overall flora itself relates pretty closely to what we see in female anatomy. With men, Their issues can arise more from being uncircumcised. The flora associated with an uncircumcised penis is going to put them at higher risk for inflammation, and that inflammation then puts them at higher risk for contracting sexually transmitted infections. All right. Urinary tract infections are 80% of them caused by E. coli from your own flora, especially for females. When we see this presented as prostatitis for men, it tends to be from flora from contact with other people. So usually sexual contact will introduce flora into the urethra of the penis, and then that biome, the introduction, I want to say biome because it's not your own biome at that point, right? You're being truly infected by somebody else's flora, and then that causing travel through the urethra and up into the prostate via that route. You could also just have urinary tract infections develop as well for males. But that usually tends to be E. coli as well. And we see this more often with unprotected anal sex, for example. Leptospirosis, this is a bacterial disease that is technically a zoonosis. We see this primarily in animals. We have shedding of the bacteria in the urine into like soil, and then you can have exposure of mucous membranes or by ingestion or by open wounds to these bacteria and leads to presentation of this disease. The leptospirosis phase, you'll have flu-like symptoms that can be pretty severe. You can have some other aspects of tissue getting involved as well, but it's when you have the immune phase that you can have long-term health effects and have meningitis and severe damage to other tissues in the body, especially overall organ function that can eventually lead to death, potentially. There are vaccines available and it is treatable with antibiotics. Vaginitis, inflammation of the vagina. Here, we're only having presentation of that for our concerns. With yeast infections from Candida albicans, this is opportunistic from your normal flora. These are not true pathogens, right? You can have this as part of your normal flora. When it overgrows because you take an antibiotic, you clear out the stadium seeds from your bacteria, the fungus are left over and they overgrow. That process is called superinfection, and that is something that's occurring from that opportunistic pathogen, then causing inflammation in the vagina. Then we have Trichomonas vaginalis, which is a protozoal sexually transmitted infection that is going to be associated with vaginitis. And then we have vaginosis, which is BV. So we have infection in the vagina, but not inflammation of the vagina tissue itself. But this can progress to inflammation of other reproductive tissues. This usually the decrease in the lactobacillus bacteria and the other bacteria will overgrow, and then that leads to the presentation of the infection. Gonorrhea, this is a sexually transmitted infection caused by the gram-negative diplococci gonococcus, which its official species name is Neisseria gonorrhoeae. Issue with this one, of course, is going to be antibiotic resistance. We often see the inflammation associated with gonorrhea making you more at risk for HIV infection, as well as more likely to carry other sexually transmitted infections like chlamydia. Chlamydia is a separate sexually transmitted infection caused by the bacteria Chlamydia trachomatis. It also causes the most common infectious cause of blindness in the world and is the most common of the reportable STIs. Now, Trichomonas vaginalis is actually more common, but we don't report it because it is just so common the CDC doesn't really track it. For syphilis, there was the Tuskegee study done in 1932 on Black men and not informing them of what was happening in the study, and then just watching the results of syphilis and how they track and progress in these men without their full awareness of what was happening in the study is very unethical. So we need to be aware of that. Syphilis is caused by a bacteria Treponema pallidum. Do know that it is a bacteria. Do know that what the name of these organisms are as well. Primary entry site of the bacteria will result in development of this chancre, and these, or chancre, these guys, depending on how you pronounce your French-based words, these are where the actual bacteria got into the skin, and the primary phase of syphilis will present with this chancre sore. It's small and hard and relatively painless, but it is the indication of primary syphilis. Secondary syphilis, we have a development of this brownish colored rash on the palms and the soles of feet and sometimes the trunk. We also have fever and lymphadenopathy, general feeling of unwellness. And the tertiary phase, we have development of gummas, which are sort of like degrading tumor presentation. So we have degradation of tissue that's involved here. So that'll be the tertiary phase, and that can develop into neurosyphilis as well, where we have dementia, blindness, and blown pupils associated with the inflammation from the infection of syphilis. Then this can become deadly. It's treatable with penicillin-based antibiotics. Congenital syphilis, here we have fetal abnormalities that can result from infection of the fetus, and we can spread the syphilis to the newborn child as well. Next, we have chancroid. This is a sexually transmitted infection caused by the bacterium Haemophilus ducreyi. We have a soft chancre this time that is actually painful and much larger than what we see with syphilis presentation during primary syphilis. So chancroid is usually associated with sex workers and uncircumcised men in the Pacific Island areas in Asia. Then we have the most common STI overall, which is human papillomavirus. These are wart-causing viruses that can cause warts anywhere on the body. When you have genital presentation due to sexual transmission of these warts, then it's true sexual transmitted infection. But we have plenty of strains of, that scared me, sorry, something fell off my wall. We have plenty of presentation of HPV that doesn't have symptoms that are apparent, unless we're doing something to investigate that further for like abnormal growth of cells and whatever that could potentially progress into cancers like cervical cancer. Most sexually active people will have encountered some strain of HPV at some point in their life. And most of those are going to be asymptomatic presentation that is cleared from the body. But again, pap smears are really important for screening for cervical cancers. We do have vaccines that are available via Gardasil that can help protect from certain strains of HPV that might put you at risk for cancers. Herpes viruses, the two that we looked at as far as herpes viruses themselves, so herpes simplex virus 1 and 2. 1 is usually associated with oral, 2 is usually associated with genital, but they can both present in either location. These guys hang out in nerves and then will become latent in the nervous system, and then will get re-expressed in any incidences of any kinds of stress. In the case of genital herpes, you have presentation of these vesicles that can be very painful to the touch, fluid-filled vesicles that this is where it's mostly contagious. When we don't have outbreaks, we're considerably less contagious, but you could still potentially be contagious. The risk with herpes for the newborn is quite severe. The virus can spread into the skin or the eyes and then be, dangerous. That way, 30% mortality rate. But once it spreads into the nervous system, we can have a meningitis develop or even encephalitis. And this can be a 50 to 80% mortality rate. So if you are having an outbreak of herpes as a mother and you're giving birth, then we will want to do C-section to avoid any risk of infection for the baby. And then group B, streptococcus colonization. This is beta hemolytic strep, and these guys are the ones that are going to be at risk for causing pneumonia and meningitis, especially in the newborn baby. So we screen mothers late in pregnancy and try to get rid of that normal, it's usually part of the normal microbiome, but 10 to 40% of women have this as part of their microbiome. And we'll close this out with antibiotics to try to prevent risk for the babies. For chapter 23, our most missed topics, we'll start with cryptosporidium. This was diarrhea illness. This is the one where the cyst is very resistant to chlorine treatment. Enters the body and survives the stomach. The cyst is very hardy and is able to survive chlorine as well as the stomach acids, and then it will turn into essentially the trophozoite forms that will cause the true infection and presentation of the diarrhea illness afterwards. And then you will shed these cysts in the feces, in the diarrhea, and then expose other people to it. And oftentimes swimming pools aren't chlorinated enough to contain those cysts and kill them off. So then you end up risking the spread of that infection through water, and the whole pool will need to be decontaminated and drained in order to make it safe again. So key symptoms, of course, we're going to have diarrhea and abdominal cramps and things that go along with that diarrhea. It's a fecal oral transmission route, but it will be presented in usually contaminated swimming water. It can be in any fresh water though. And they're very resistant. So here our memory device here is crypto can cling to chlorine and cause chaos in the colon. Speaks for itself. So which of the following is the primary mode of transmission for cryptosporidium? We know this is a fecal oral route. So is it airborne droplets? It's not going to be that one. Is it contaminated water or food? It can be food. We didn't mention that necessarily, but we know it can be contaminated with water. And if it's fecal oral, of course, it could also be food too, if you're touching the food, contaminating. Insect bites, it wouldn't be that one, nor direct contact with soil. Here it's fecal oral, so there does have to be ingestion of the cysts in order for it to transmit into the GI system. So B is our answer here. Why is cryptosporidium difficult to eliminate? Does it form spores that are highly contagious? Does it produce toxins that persist in the water? Do its oocysts, or just cysts for our purposes, if it was on our test, are they resistant to chlorine, or does it survive extreme heat and boiling? Well, our risk here is just with water. We're not boiling the water first or anything. It is chlorinated to protect us, but these guys are resistant even in that. So our answer will be C for this one. These are not spores. All right. So endospores, remember we have endospores. These guys are for bacteria only. Cysts are what basically are endospores, but these are for protozoa. So they have their own term. These are our hardy resistant structures for the protozoa. And we're not talking about toxin production or anything like that as far as what makes them difficult to eliminate from the water supply, right? Okay, moving on. Another misconcept is going to be Hep A. Hep A, again, this is going to be a virus that can cause inflammation in the liver, but we do not have chronic presentation of liver disease. We don't have cirrhosis. We don't have cancer or long-term effects of liver damage as resulting from this. It's usually actually pretty rare to even see jaundice associated with this. Only 10% of cases even have jaundice. So you'll have typical illness symptoms, sometimes jaundice and dark urine and pale stool that we see associated with some liver inflammation. And symptoms usually last about 2 to 6 they develop about 2 to 6 weeks after exposure and can last for weeks to months, but most people recover fully without lasting liver damage. It's a fecal oral route of transmission, usually from contaminated drinking water or food, close personal contact, or poor hygiene or sanitation. It's resistant to desiccation and certain disinfectants. We do have vaccines that are available for this, and more commonly to prevent this will result in preventing this via normal hygiene practices of preventing any sorts of fecal oral transmission route types of diseases. Hepatitis A, to help you guys remember, A plate, you get Hep A from A plate of food and water, for example. And you can avoid with a vaccine. Hep B and C, though, these are blood contact. B and C, blood contact. And C can't be curbed by a jab. It can't be, there's no vaccine for C. So just to summarize that. So what is the primary mode of transmission for hepatitis A? A plate. That was our food and water one. So is it blood and body fluids? That's not the primary means. Airborne droplets, we didn't talk about that. We talked about fecal oral route through contaminated food and water, and insects don't play a part in this. And which of the following is true about hepatitis A? Does it cause chronic liver disease? It does not. We know that you can clear this virus and then recover without lasting liver effects. Can we prevent it with a vaccine? We can. Is it transmitted through blood and sexual contact? Not primarily. You could potentially spread it this way, but its primary mode of transmission is fecal oral. Is it commonly treated with antiviral medications? If you really needed to treat this with an antiviral medication, there probably are ones that could help clear the infection, but not commonly. That's very not common. Most of the time, more commonly, your body will just clear it on its own. All right, next for chapter 23 are our schistosomes. Schistosomes are helminths, but what kind of helminth are they? They are flukes. Flukes are part of the flatworm. So we're talking about helminths then platyhelminthes, which are our flatworms. And our flatworms are divided into the cestodes, which are our tapeworms, and then the trematodes, which are our flukes. Okay, so trematodes. So these guys, they get in through the skin, causing the swimmer's itch. There's usually an intermediate snail form that you guys don't need to know about this, but he's also known as like a snail fever or something like that. Anyways, you get the itching in the skin, and then you'll develop symptoms throughout tissues throughout the body as these flukes spread throughout the whole body via the blood system. We get hepatomegaly, so enlargement of the liver, and splenomegaly, enlargement of the spleen, and other tissues can become involved as well. They can cover themselves in our own antigens to hide from our immune system, and damage can be chronic and lasting as you carry the infection. Again, usually have an intermediate of snails in these guys. So you want to avoid contact with any contaminated water, control snail populations in areas of risk, and, treat with praziquantel as your anti-helminthic drug for this one. So to remember, schistosomes swim, stick, and settle as skin-penetrating snail scent flukes. So just to help you guys remember how they get in, how they infect in their life cycle, and what makes them different. So schistosomes infect humans. How do they do this? This is our swimmer's itch thing. So it's getting in through intact skin from freshwater exposure to contaminated water. So are we ingesting them through the food? No. Are they penetrating our skin during contact with contaminated water? Yes, that's it. What about the others? Do we inhale them? No. And do we have direct contact with infected animals causing the spread? No. So just B is our answer. What type of organisms are schistosomes? They're helminths, but flukes, roundworms, and tapeworms are all helminths. So we can already rule out protozoa. We talked about how these guys are flukes, right? And I would also keep in mind, we learned that the terminology for flukes was trematoda. So trematodes. So that's like this, or trematode. I don't care how you pronounce it. Okay, remember, tapeworms are going to be your cestodes, or cestoda is the group, and roundworms are nematodes. So don't forget those terms. For chapter 24, our first most misconcept, HPV is the most common sexually transmitted infection by far over any of the other sexually transmitted infections. Human papillomavirus is a virus that can cause genital warts. There's very low risk strains that cause that. The high risk strains, we have risk for cancer development, that these guys usually have no other symptoms that are associated with them. So it's very common, it's very contagious. It's sometimes hard to even know that you have this. You don't have presentation of symptoms, so you're less likely to reach maybe for protection if you're with a partner that you trust, but you want to be sure there's no usually presentation of symptoms if you are a carrier of the versions that can cause cancer. There's no cure for these infections, but the body's immune system usually will clear the virus out. There are vaccines to prevent the more common strains that are available. And so you want to use condoms to prevent as much as you can, and that will prevent spread of the infection. And you also want to go through regular pap smears to screen for the presentation of the virus to see if you are carrying it without any symptoms. You can have positive presentation. You could have like a pap smear that pops positive for certain HPV viruses and not have any abnormal cells or anything whatsoever. But it does let your doctor know they want to continue to look out for progress of that disease and make sure that your body is clearing it as opposed to forming any abnormal cells. So HPV, hiding in plain view, pass through passion, it's the most popular STI. How is HPV primarily transmitted? Is it through the air, by contaminated food and water, through skin-to-skin contact during sexual activity, or by sharing personal items like towels? Most commonly, it's primarily transmitted through sea. Sharing personal items, it could potentially allow for transmission of these things, but especially when we're talking about the cervical cancer versions of these guys, using personal items for those strains aren't as commonly going to be able to transmit it. If we're talking about the wart transmission, these guys don't do very well outside of the body. So unless you're using it just directly right after that other person, you're not really going to be at risk for that. So it's not as much of a concern, not by far. Which of the following is true about HPV infections? Do most of them cause immediate symptoms? Do all of them lead to cancer? The majority of HPV infections clear up on their own without causing harm, or HPV can only be spread through vaginal intercourse. So this answer is going to be C. Most of the time it clears up. Not every strain will lead to cancer. You can even have the cancer strains and still clear them out just fine from your body. In fact, most of the time you will, but you still want to keep an eye on that for safety's sake. You can get it from exposure, not just through vaginal intercourse, but through any skin-to-skin contact, really, honestly. All right, the stages of syphilis is our next most misconcept. So remember, primary stage, we're going to have that chancre, where we have that painless sore, small, hard, painless chancre, where we have the site of entry of the organism. Secondary, we have our flu-like symptoms, as well as mucus patches in the mouth, which we didn't talk too much about in class, but it's a possible side effect. But more commonly with our secondary stage, what we're looking for is that rash. It's brownish rash on the palms and the soles of the feet. You can become latent after the primary or the secondary stage, where you just stop presenting with symptoms for a long time and can carry the bacteria and still be contagious with it. But you can also develop into tertiary stage, which is late syphilis, where we have gummas developing, which are soft tumor-like growths, where we have degradation of the tissue that look often like open sores or wounds. Then we have the neurosyphilis, where we can have blindness and neurological issues and lead to dementia and even death if it's not treated. We treat this with penicillin-based antibiotics. Earlier treatment, the less likely you'll have any lasting damage or sequelae from the disease. I dropped my pen and I don't know where it went. Here it is. And there's also, of course, that risk of the congenital syphilis and leading to abnormalities in stillbirth and miscarriage as well during pregnancy. This is caused by the bacterium. Remember, it's bacteria, Treponema pallidum. So silly people leave terrible scars is reminding you about, it starts off as just a simple sort of sore that might just seem like a silly little sore at first, but you could have progression into this terrible disease of syphilis. It's transmitted from person to person and can have terrible sequelae associated with it. What is the characteristic sign of syphilis in the primary stage? Primary, we're looking for that sore, that initial chancre sore. So is it a rash in the primary stage? No. Is it a painless ulcer called a chancre? Yes. Do we have swollen lymph nodes? We see this in secondary. Mucus patches, also seen in secondary. Rash is also secondary. What stage of syphilis is characterized by flu-like symptoms and the rash, and then the mucus patches? We kind of already answered that with our other question, but this is going to be our secondary. Remember, primary, we have that chancre. Latent phase, this is like literally latency. You have no obvious symptoms or no progression of symptoms. So it's just hiding out. And the tertiary stage, this is where you're going to have presentation of gummas and then neurosyphilis. And progression can lead to death. All right, now we have chancroid. This is a sexually transmitted infection that's going to have a chancre as well. It's a larger sore than what we see with syphilis. So it's a larger chancre. It's a soft one. It's kind of weepy and mucousy. And it can be very, very painful. So this one isn't a painless, tiny little sore. This is a big, painful chancre in this case. It's caused by the bacterium Haemophilus ducreyi. It's going to spread through sexual contact, and we have transmission through, again, usually the genital contact. And you'll have that painful ulcer, often with a much larger size, and it's much softer than what we see typically presented with like the syphilis ulcer. It's very painful. You can also have very painful and swollen lymph nodes that will develop as a result of having infection with this bacteria. And this is treatable with antibiotics. It's usually associated with sex workers as well as being uncircumcised. So here again, Haemophilus ducreyi leads to chancroid, and we have that soft, large, painful chancre with this one. So Shansies hurt, hard to miss, Ducreyi's ulcer, and dreaded buboes to help you remember the presentation of this disease. The buboes are those swollen lymph nodes that can present. We didn't talk about those in our lecture, but if it helps you remember the difference between this and syphilis, whatever you need to remember. All right, what is the primary symptom of chancroid? We have that painful genital ulcer. You could also have eventually, again, swollen lymph nodes developing, but that painful genital ulcer is the key for this one, okay? There's no rash associated with this. You're not going to have flu-like symptoms or fever or itchy vaginal discharge. You're going to have this painful ulcer as our primary symptom. What bacterium is responsible for causing chancroid? This is not Treponema pallidum. This one is syphilis. It's not Neisseria gonorrhea. That's obviously gonorrhea. And it's not going to be chlamydia. Chlamydia trachomatis causes trachoma, which is the most common infectious cause of blindness in the world, or just proper chlamydia, which is our STI. So our answer is Haemophilus ducreyi. So that is it for the review for those chapters. So I'm going to close out of this. And we're going to pull up our review. Maybe. Here we go. For the final. And we're going to go through our topics for units 1 through 3 for this part of the rest of our review. So we'll start with unit 1. So I'm skipping over chapter 1 and moving into unit 2 or chapter 2 because chapter 1 was really just like basic intro stuff. There are some terms and stuff that you might want to review for that, but a lot of that was just introduction to stuff that we have used continuously throughout the semester. I'm not going to touch on it. That'll be on you guys. So we'll go over our major topics here. I'm not covering everything. I'm going to reiterate, this is not comprehensive, not. Okay, this is not going to cover everything you need to know for the exam. Your study sheets have everything you need to know for the exam. I'm going to cover some of the bigger topics here to refresh our memories on them. So with chemistry, remember about how atoms make up all matter, and there are the different bonds that can happen between the atoms to make up molecules and compounds that those bonds dictate essentially how those atoms are going to behave between one another. And those often create how these molecules will behave, on a larger scale for a lot of different biomacromolecules and the different ways that our metabolism and everything work together. So what I'm going to get here are 3 major types of bonds that we learned were covalent, ionic, and hydrogen. All right, so our covalent bonds, we have proper sharing of electrons. Ionic bonds, we don't share the electrons. We have donation. So one atom gives its electron to the other one. Donate or transfer, however you want to look at it, but we're not sharing them anymore. We've actually given an electron to somebody. And now that sets us up for that whole concept of what we talk about later on, where we talked about oxidation and reduction, where we have losing electrons is oxidation and gaining electrons is reduction. Leo says GER, or if you like oil rig better, either way. But ionic bonds absolutely have to do with that every time. If you have an ionic bond, you have oxidation and reduction going on. And then we have hydrogen bonds where we just have to like hogging electrons. But we're still, one atom hogs the electron, giving the other one, usually our hydrogen, a positive charge. Right? So hydrogen has a positive charge. It's not been ionized yet. It still gets to see that electron every once in a while. It's still part of that covalent bond to the other more electronegative atom, but now has a positive charge. And this hydrogen having that positive charge will interact with things that have a negative charge, like maybe other oxygen atoms that have a negative charge. And this here would be your hydrogen bond. Okay. So it's of another molecule, and these charges, these polarities are what are governing things. So a hydrogen bond itself between that positively polar hydrogen and a negatively polar atom of something else that are attracted to one another. So we're talking about attractions guided by the fact that the hydrogen has that positive polarity. That's why it's a hydrogen bond. Okay. How do molecules interact with water, and what are some examples that we've discussed? Here, this is where we're talking about hydro. You can be phobic, water-fearing, philic, water-loving. So if you love water, you are polar typically because water has polarity. Right? Due to the fact that oxygen is electronegative, it hogs. I don't know why it has 2 G's now, but it does. It hogs electrons, leaving these guys with positive charges and this guy with a negative charge. And those guys can interact with other polar things. So things that are hydrophilic tend to be polar. Things that are hydrophobic tend to be non-polar. Right? And then we have amphipathic, where you will have both non-polar parts and polar. So you might have a long hydrocarbon tail, and at the end of it, you might have like a carboxyl group or something. And then these oxygens having that charge on it. Or it could have a phosphate group. We've talked about the heads of our phospholipids. So the phosphate groups are very highly negatively charged, and the tails are very hydrophobic. That's very characteristic of our phospholipids that make up our lipid bilayers, and why lipid bilayers are characterized, structured the way that they are. Very important for how all those molecules function with one another. All right. How do we measure pH? pH is measured as the negative logarithmic of hydrogen concentration, which means that high number for pH, so if we're talking 14, equals low H plus, because this negative here inverts the scale. So the more H plus, 0 would be a low number, and that means high concentration of H plus. So this would be acid, and this would be base. And we know that 7 is neutral. All right, what makes a molecule organic? Carbon-hydrogen backbones. You can have carbon, but you also have hydrogen there to support it and create those stable interactions with the carbon. So we're talking about carbon-hydrogen backbone. Large molecules in the parts here we're talking about our biochemical macromolecules. So our proteins that are made of amino acids. Our nucleic acids, so like DNA and RNA. These are made up of parts that we call nucleotides. And these nucleotides, we remember they consist of a 5-carbon sugar. So it's an ose. So it's the ribose or deoxyribose. It has a nitrogenous base. So nitrogenous, literally talking about nitrogen being part of it. And then we have a phosphate. That's phosphate. So that's a nucleotide. We also have carbohydrates. Those are going to be made up of monosaccharides or single sugar units. And then we have our lipids. And our lipids are made up of fatty acids and long carbon hydrogen chains. And it just kind of depends on what type of lipid you're talking about. This could also be the sterols like cholesterol that is important for creating structure in the lipid bilayers. For protein structure, you do need to know the difference between primary, secondary, tertiary, and quaternary protein structures. Your amino acid sequence just straight up, whether you have proline, alanine, phenylalanine, methionine, cysteine, all of these 20 different amino acids that we know of, the sequence of those guys makes up the primary structure. The secondary structure, those different amino acids being next to each other cause structural changes from just being next to each other that develop into beta sheets. So those are beta pleated sheets that are literally just straightaways going back and forth, or alpha helices, which are literal helixes, like twists and turns in the structure of that ribbon of the amino acid sequence of how those amino acids next to each other are interacting with themselves and their environment. Then those guys can interact with amino acids well across the way as we're starting to fold up, from hydrogen bonds, from polarity, from interactions with pH in the environment, from creating disulfide bonds with other amino acids and what have you, leading to the tertiary structure. So now we're tying up that ribbon, and the quaternary structure is bringing several ribbons together to make a functional protein. For chapter 3, we had our 5 I's that we were involved in. That's talking about basically how we're going to be investigating our microbes. In our 5 I's, we have inoculation, we have incubation. So inoculation, we're just introducing the microbe to whatever the media is. For incubation, we're growing it. And then we have isolation. We're literally just isolating a specific pure culture. So we're creating pure cultures of individuals. Then we have inspection, where we're going to be doing testing on them, and then identification based on all of these results. The different types of media that we talked about selective media that selects for certain species. So it usually has something in it to prevent growth of other species. We have differential media that tells the difference based on color changes, usually that are caused by metabolic byproducts. We had enriched media that would have introduced growth factors that are necessary for fastidious bacteria or other microbes that are fastidious. They require these growth factors. They cannot make them for themselves with bare constituents. So they have to have them supplied in their diet. Growth factors need to be supplied to fastidious organisms. The media that they grow on that contains the growth factor is called enriched. Then the isolation methods, we talked about creating pure cultures. We talked about streak plates, so the quad streak plate. I'd still remember about that one if I were you guys. That's a pretty simple concept. You guys know how that works by now. Then we talked about microscopy. There were a couple old guys that we learned about. This is that basically were involved in discovering microscopes and how they work. And you guys don't need to know about these guys. That's why that's on there. That was an old slide. But you probably should know how microscopes work, right? Know the basic parts of them, like we did for the lab. Know what magnification steps are going on. We're creating the real image from the actual initial light source contact. So the light through the specimen contacts our objective lens. We create the real image. It hits some mirrors, goes through the optical lens, creating the virtual image. Oil is still used to help decrease light scatter and increase the clarity of the image so we can see more detail in our images. Then we have stains that we use to create contrast as well as to differentiate between different structures. Still be aware of those different stains, like the endospore stain tells you that there's endospores or not. We'll talk about the endospores in the next chapter, but and all of that stuff. So chapter 4, we learned about bacteria and archaea. Both of these guys are eukaryotes. They do not have a nucleus. and therefore do not have organelles. Organelles are membrane-bound structures that have specific purposes in our eukaryotes, but our eukaryotes do not have them. All of those processes can still happen in an eukaryotic cell. They're just not being compartmentalized. So keep that in mind. A lot of times, if you need a membrane for a process to occur, like aerobic respiration, photosynthesis, or anything like that requiring proteins to be bound to a membrane, that's usually happening on the inner membrane, that cytoplasmic membrane of these bacterial cells. Okay. Gram-positive means you have a thick peptidoglycan layer. Peptidoglycan is our major component of the cell wall for bacteria. Gram-negatives have thin peptidoglycan layers and an outer membrane. It says others because there are other bacteria out there that don't have any cell wall. And we have archaea that they have their own cell walls and membranes, but they aren't composed of the same materials. They aren't peptidoglycan or anything like that. They're their own thing. And so that brings us actually down to that bottom, the others does, down to the cutes. Remember, we had the firmicutes, the firm cell wall of the gram-positives, the gracillicutes for the gram-negatives. We had the teneracutes for our ones that lack a cell wall. Think of tenor, okay, teneracute. And then Mendozacutes. Joey Mendoza was my old boyfriend. That was a long time ago. Like that was archaea. Okay. Mendozacutes, archaea. So our cell shapes and arrangements, again, this should all be review. We have our cocci that are round, our bacilli that are rods. We had arrangements of our staphylate being our grouping of our irregular bunches or grape-like clusters. Strepto was our chains. Palisades were the upright rods kind of next to each other like fence posts. Then we have flagella and how bacteria move with their flagella. You can have more than one flagella. They could be at the ends of the flagella or all over the flagella. Our terms for these guys, if they're all over, it's called peritrichous. If you have one flagella, you are monotrichous. If you have tufts of flagella, then you are lophotrichous. And if you have a flagella at each end of the poles of the rod, then those are amphitrichous. Again, the flagella move in a rotating fashion. They do not move in a whip-like fashion. The flagella for the eukaryotes, like our protozoa, they move in a whip-like fashion. So it's very different. Flagella for bacteria rotate. And so they move in runs and tumbles. Runs shooting straight away by rotating the flagella. When you stop and rotate the flagella in a different direction, that's a tumble. And that's how they will change the direction. And they will combine runs and tumbles to try to orient the cell in certain directions, either towards a chemical signal or away from a chemical signal for chemotaxis. If it's light that's getting the signal, then that's phototaxis. Capsules are sugar or protein-based coverings on the outside of our bacteria that can allow for adherence as well as evasion of phagocytosis, which is when our white cells are going to eat up those bad guys. It's harder for them to grab onto a capsule or a slime layer, which is just a loosely associated version of that. These capsules and slime layers aid in the creation of biofilms as well. Biofilms allow these bacteria to sort of live in a colony with one another of different species, though. So typically a colony is one cell that grew up and multiplied, right? But biofilms, we have all different kinds of species growing amongst one another in the same grouping, creating a literal film. And they can send chemical signals out that the other guys can interpret to let them know about the health of the biofilm and whether they want to keep participating in it. And that's called quorum sensing. Endotoxin is also LPS or lipopolysaccharide. So it's multiple sugars with fatty acid component on the surface of the outer membrane of gram-negative bacteria. That's endotoxin or LPS. It's all the same thing. LPS, endotoxin, lipopolysaccharide is the same molecule on the surface of gram-negative bacteria. It can trigger immediate recognition as this is a bad guy and shouldn't be in the body, and therefore is a PAMP or pathogen-associated molecular pattern, which we'll talk about in later chapters. But when we've learned about it already, but we'll review it again. So that's endotoxin. Gram-negatives only, okay? Endo, because it's part of the gram-negative cell. It's not a toxin that they secrete. That would be an exotoxin. Plasmids are extra bits of circular DNA that have extra genes on them for either making toxins or creating antibiotic resistance or whatever. Plasmids are not required for survival. So bacteria can survive without the plasmids, but the plasmids can provide genes that help the bacteria survive in different conditions, like presence of antibiotic or something. So they can be helpful. Endospores are hardy and dormant structures that some bacteria, the Bacillus and the Clostridium species, can make. So those are gram-positive species. And those endospores are essentially the hardy ability. So they are not going to be metabolically active. They're literally just protecting the genetic information until conditions improve. Once they improve, the endospore will create the vegetative cell, go through germination and germinate like you would have a seed that was a plant germinating into a plant. And then that becomes a vegetative, active, metabolic, dividing, reproductive form that we're used to talking about with bacteria. That's the vegetative cell. We talked about the cutes already. I will say one thing that isn't on here that I'd like to mention is just the acid-fast aspect. So remember, the acid-fast stain, you don't need to know the steps of the stain for the lecture, but you need to know that acid-fast stains are positive for mycobacterium species like tuberculosis. They have mycolic acid on the surface, and then if they have mycolic acid, they are positive for the acid-fast stain, and that mycolic acid makes them more resistant to certain environmental factors. Chapter 5, we have the endosymbiotic, eukaryotes, right? So eukaryotes are any organisms that have a true nucleus. That's what the word literally means. It's true nucleus. And if they have a true nucleus, I forget that doesn't erase when I do that. And they'll have organelles. And these are membrane-bound structures that have function in the cell. We've compartmentalized functions of the cell in the eukaryotes, okay? So at this point, like this says, please tell me you know what a eukaryote is. Please at least tell me that you can tell the difference. You know the basic difference between a eukaryote and a akaryote, having the nucleus and or other membrane-bound structures, okay? The organelles and, the endosymbiotic theory states that these organelles, these membrane-bound structures, may have evolved from a symbiotic relationship that developed between like a cell that ate up another cell, and then they got benefits from one another, and then eventually over time those became the organelles. There's evidence that supports that as far as chloroplasts and mitochondria go, because chloroplasts and mitochondria have their own DNA, their own ribosomes. They divide separately. They have two membranes, just like gram-negative bacteria have, and they have circular genomes of their own. Remember, the genomes for bacteria are going to be circular in nature, whereas eukaryotes have linear chromosomes. Organelles and their functions here at mitochondria create the energy. The nucleus houses the genetics and is basically like, the like memory storage of the cell. So it has all of everything the cell needs to do coded in the genetics within the nucleus, right? And all that DNA only ever stays in the nucleus. That DNA never leaves the nucleus unless we are making new cells during mitosis. So we have the nucleus, we have the nucleolus, where we're creating ribosomal RNA. The ribosomal RNA creates the ribosomes that are going to eventually read the messenger RNAs to make the proteins, right? So those structures are very important, like machine-like structures for later on. We have the nucleus, we have the endoplasmic reticulum. That is another membrane-bound structure where we are studied with ribosomes for translation to occur, so we can start making proteins in the rough endoplasmic reticulum. The smooth endoplasmic reticulum will add on other molecules that are non-protein molecules. Then we have the Golgi apparatus, where we have packaging and transport in vesicles, including creating things like lysosomes packaged with things like lysozyme that break down peptidoglycan, acids, oxidative things like that will actually be creating these problems. This thing keeps saying that people are joining my meeting. Did I start like the wrong meeting or something? Hold on really quick. Zoom. It's not telling me. Yeah, I'd create a freaking different meeting. Thanks. Hold on.